There are, I think, two basic problems with this. First of all, we're not looking for investments, because this is not a company, this is a project that was mentioned earlier on that has stemmed from research at Karolinska Institutet, is now in collaboration with us at Karolinska Innovations, as well as Invent2, which is a tech transfer office in Norway, and the researchers both actually in Oslo and in Stockholm. So he has funding all the way up to phase 1 actually, so we're not looking for money for the moment, but this is more like an example of what we are developing within the innovation system in Sweden, in Karolinska, and what sort of research that comes from here. The second problem is I'm not an expert in immunocell therapy, so if you have any very, very tricky questions on that part, I can point you in the right direction. So what we're trying to... Nothing happens with this one. Oh, sorry. That was the right one. So what we're trying to develop is a targeted a therapy, immunocell-based therapy towards hematology malignancies. And there is a need, of course. I mean, we know that all patients with AML and ALL are not treated. This is the 5-year survival rate coming from American Cancer Society. But we also see potential for other therapies such as neuroblastomas and melanoma. So we believe that the potential number of patients that will be treated in Sweden, and now we also count the ones that we are actually going to treat, not the total amount of patients in Sweden, and Norway will be about 700 per year. So there is a definite need for that. And this is based on natural killer cells. Natural killer cells were found in the mid-70s in Karolinska actually, and it's been explored in different ways in the clinic, and they are cytotoxic lymphocytes, which has both inhibitory and activating receptor controlling their activity. And upon transformation, for example in a tumor cells, very often one see a loss of MHC class 1 molecules, and then one see an activation of the cells, and they will kill the transformed cell. This can also be achieved, of course, if you take cells from one patient or a donor or give it to a recipient where there is no match of MHC class 1 molecules. And that's, of course, another way of doing that. And NK cells has been tried in the clinics, but hasn't really taken off yet. We believe that we represent the next generation of targeted immunotherapies, because we think that we can sort of get the cells that we want actually. And in the donor situation, the donor has a population of different NK cells, but only a few ones having the desired specificity. So it's the red one that we want. But there's not enough to be able to sort of kill enough cancer cells in the recipient. However, when we treat the cells, we can stimulate them, so the subpopulation that we want is expanded. So we get more of them. So we get more of the red cells, basically. We get more of the cells that we want, that we're going to kill the cancer cells. And we have a patent file on this procedure. Is a PCT phase right now. And there are also some publications out. What we think that we can get, which is different from the other NK cells therapies being used, is that we get an enhanced cytotoxicity. We also can predict the specificity of the cells that we get, so we can sort of target them to the right cells. And there's also no genetic manipulation of the cells, which there is in some other protocols. And this is just showing some data that actually going from about 10% of the cells we want, after about two weeks we get 70% of the cells that we want from the total population. And they are also more, what is called educated, is more prone to kill the target cells actually. So they are more active. Not only that, with more of the cells, they're also more efficient. This is just showing an example that actually it works to kill the right cells. So this is primary blast from children with ALL. And this particular cells has a C1 background. That means that these cells should not kill them because they are sort of recognized self. But this one has another set of MHC class 1 molecules, so there's a missing self. And there we see that we kill more than 80% of the cells in this experiment. So we have shown that it works in vitro. I mean, of course, we're in ex vivo. Of course, the question is, does it work in vivo as well? And we'll see that in a couple of years. The type of products that we think that we can develop within this project is, first of all, we can of course do the whole procedure in the clinic, and that's of course how we're going to start. And actually the researcher is currently doing these type of trials, but with another protocol in Karolinska Institutet or Karolinska Hospital. So we sort of have the capacity and the knowledge to take it all the way to the clinics. In the future, we're going to try to develop more off-the-shelf products, such as GMP cell lines as well as bead products that will stimulate the cells to get the required specificity that we want. So we can actually not only do it to the limited number of patients, but actually sell it to the rest of the world and save some lives and make some money. As I mentioned, we have a fully funded development pipeline. So we have research funding in the order of 20 million Swedish kronor, and also we have some commercial funding in the order of 5 million Norwegian kroner coming from Biotech 2021 program. And what we're focusing on right now is to developing the in vitro beads as well as the cell lines. And of course, in parallel, we're testing that in primary cells coming directly from the clinic. Then we will, of course, test this for efficacy and safety in animal models, and of course do all the requirement tox studies so we can go into humans, which we'll think will be some experimental trials in the beginning of 2017. In parallel with that, we're working with the IPR, of course, and the commercial aspects. We're currently not looking for a partner. We think that we will need a partner in about a little more than 2 years. But we, of course, we are opportunistic. So if we stumble upon someone that can complement the technology we have, we, of course, are interested. And we have been talking to potential partners. But surprisingly enough, the big pharma thought was a bit too early. Has anyone heard that before? The team that we have, so the scientific team is led by Kalle Malmberg, who is a MD, PhD, and professor both at KI and in Oslo, at the Oslo University Hospital and Oslo University. He, together with Viviane Bessiat, who was his postdoc, are the inventors of the technology. Viviane is now back in Paris pursuing his career. As a scientific support, we also have Hans Gustav Ljunggren, who is a professor out in Huddinge and also Dean of Research at KI, but has an enormous knowledge in cellular and molecular immunology. The commercial part of the project is headed by Invent2, which is the tech transfer office at Oslo University and Oslo University Hospital, and is, at least in my mind, the best TTO in Norway. And of course, we have us doing this. And I should just mention a little bit what we are doing in general, the sort of the task for KI Innovations. So the overall task of the whole innovation system, whether it's sort of the non-commercial part or the commercial part, of course, create value from research at Karolinska Institutet. And the innovation system spans all the way from education and research up to companies on the stock market. And we create value by identifying interesting projects, and we do it partially by actually being in the scientist in the different interesting areas such as cancer, neuroscience, and diabetes, where we have innovation managers working closely to the scientist. We evaluate the projects, we verify them, and that we do with some support from Vinnova, the VFT money that we can help the scientists. And we help them develop the projects and also, as you've seen in this case, partner with relevant actors. And you will see a later case later on today, Apriya, who has actually come from our system, now is a partner with Karolinska Development. As a summary, I'm trying to convince you that we're trying to develop a new NK cell-based therapy, which give enhanced toxicity and predict the specificity. We think we have a strong commercial and scientific team. We have research and soft money all the way to phase 1, and we will need a partner in about 2.5 years. So thank you.